Hello. Yeah, cool. Well, yeah, good morning, everyone, and thanks to the organizers and Esther for having us here. Um, so I'm going to be talking about maglev, which is this recent sort of development in fluorescent proteins, and we think perhaps generally magnetically controllable proteins. But first, I wanted to start with kind of where our group comes from, which is this field of synthetic biology. So this is about like applying engineering principles to biology for all sorts of purposes across, I guess, fundamental science, developing biotechnological tools, but also across medical and agricultural. Um, and in engineering, it's sort of driven by this idealized cycle of design, build, test, learn, where you kind of make something new and then you build it and then you see how it performs and um, you take those learnings to kind of iterate and develop some kind of technology. And in the past few decades, this has been applied in molecular biology, as we heard a bit of the history before, in some really powerful ways. And in one in particular is with this green fluorescent protein. So this story actually exists throughout biology, but this is a particularly, I guess, prominent one. Um, in jellyfish, they kind of glow green, and this was realized at some point that you could isolate the protein that is responsible for this green glow. And actually, it was only kind of a few years later that people realized, oh, having a protein that like glows green is very useful. And it turns out you can use this across all sorts of um, biological assays because you can target things that you're interested in, you can label certain things. Um, and GFP actually, the protein that is in the jellyfish is not the same as the protein that is in use across basically every biology lab today. It's seen kind of a decade or so of development, so various attempts to make mutations to make it brighter or more stable, either through evolution or through rational design. Um, and so I think with the kind of proteins that we're going to present today, we kind of see it as being at the start of this paradigm. So again, we have like a new sensor, perhaps fluorescent marker, and it's probably going to take some engineering to get it to the point of being widely adopted, but we think it has some promise. So yeah, so GFP in this, every tool, I guess, has its trade-offs. GFP is very good spatiotemporal in that, you can see where it is and when it turns on or when it's expressed. The experiment is pretty easy. Any kind of undergrad molecular biologist can get GFP to be expressed in some cells. It's genetically interventionist in that you need DNA. I guess the tool that we're going to talk about is also genetically interventionist, but perhaps overcome some other challenges. So maglev is this protein that was reported maybe a year ago now from Andrew York and Maria, folks at the time at Calico in the US, and they've since started a company based on some of this work and also moved to different research organizations. Like GFP, it has this interesting origin story where it comes from oats, although in this case, the oats weren't already glowing and magnetic. It's just this protein kind of is a love binding, which is useful for photosynthesis. And that seemed to be a good starting point. And so they engineered this protein. So this is not the natural protein in oats. It's had some changes to a point where you can see these very large magnetic field effects in the fluorescence. So you turn on a magnetic field and the fluorescence goes down. You turn off the magnetic field and it goes back up. And it's a massive change in fluorescence. So this is what was really new about this compared to something like the cryptochrome, which also exhibits, magnetic field effects, but none so large and also not so kind of robustly that it's a harder protein to express and get working, for example, in bacteria. So as a biotechnology, this was very exciting. And then also at the time, because of the way that this was developed, there was actually the question of what is going on. Is it the radical pair mechanism, which I'm going to kind of refresh people on or explain what that is for the first time? Or is it something else? It could be a conformational change. Kind of, we didn't know to begin with, and we've now done a lot of follow-up experiments where we've established, I think we're pretty confident it is likely the radical pair, or at least certainly some spin-based mechanism. And so, in fact, it wasn't just this one protein. There's this whole class of proteins now where they are all responding to magnetic fields. Later, Harrison will present some other works that have come out of other groups, but Even just from the US group, there was already GFP with flavin, red proteins with flavin. So this is kind of emerging, I guess, as a general class of proteins that you can make magnetically responsive. So far, all fluorescent. So the radical pair mechanism, from a bird's eye view, is this idea that you have a photocycle and some other situation going on in a kind of dark pathway. And basically, over on the right, we have this spin triplet mixing, which I'll go into a bit more detail. But the point is that magnetic fields can influence this right-hand pathway. I guess it's, yeah, this right-hand pathway. And that changes the ground state repopulation rate. So when we do fluorescence measurements, what we're really measuring is kind of like indirectly how quickly is the ground state being repopulated. And in the diagrams I'm going to show next, I'm going to kind of get rid of the photocycle. But just so that that's clear, we're not measuring like fluorescence on the right-hand side. We're measuring fluorescence on the left-hand side, which is this measure of the ground state, which is an indirect measure of what's going on in the radical pair. So the radical pair mechanism always goes something like this. Light comes in, there's perhaps an intersystem crossing or not. In our system, there is an intersystem crossing. So we go from a singlet excited state to a triplet state. This forms a radical pair. And that radical pair kind of has two possible states, singlet and triplet. And importantly, those two states have different fates. They have different chemical fates because the singlet can much more easily recombine back to the ground state, which is also singlet. Whereas the triplet has to go through some kind of pathway if it's going to get back to the ground state. Could also be lost, for example, as a signaling molecule. This could be a way that magnetic field effects influence downstream biology. But basically, the physics of the singlet triplet mixing gets converted into chemistry in this molecule that kind of has different pathways it can go. And so the reason that there are triplet and singlet states is something like this. So we have two electrons and they can pair up in different ways, either with spin 0, which can only be a projection of spin 0, or spin 1, which can have 3 different projections. And in the absence of any magnetic field, because of kind of hyperfine couplings around, these states can all kind of mix together. And so overall, you get some average rate of singlet and triplet back to the ground state. However, when you add a magnetic field, there is Zeeman splitting on the triplet states, and that separates them energetically. And so in our case, because the system is triplet born, so that there's this very fast intersystem crossing from the excited state singlet into the triplet state, this means that mixing can no longer occur as much. And so basically the system is more so locked in the triplet state. And as I said before, the triplet takes longer to get back to the ground state. So we have this slower repopulation of the fluorescence ground state, and therefore we have a slower fluorescence photocycle. And so if you can separate the energy levels, you should be able to reconnect the energy levels as well. Using radio frequency energy, because that's like what the energy gap is. And so this is really the experiment that we did, which established that this is the mechanism rather than something else. Could have been anything, because the way that these proteins were developed was through directed evolution, wasn't through like rational engineering or necessarily like knowing what's going on. Obviously, there was a guess that this is probably going to be the case, but these proteins came about with no necessary mechanistic. And so we did this measurement where you apply a static magnetic field, and I'll just show here. So Zeeman splitting means you apply the magnetic field, and that sets the energy gaps between the triplet states. So you know exactly what that energy gap is, and therefore you need to apply on resonance microwave signal to kind of exactly match that energy gap. So in this case, we have set it to whatever it needs to be for 600 roughly megahertz. And we saw a really nice ODMR peak. And this is quite exciting when we saw it, because this is in a single bacteria that is in very kind of normal culturing conditions. So it's alive, it's at room temperature, there's no like oxygen, like it's just on a microscope. And that's great because in physics, actually, it's kind of hard to come across systems that exhibit something like ODMR just at room temperature. So there's the NV center that we've heard about or seen a bit about. And like that is a fantastic platform because it is so robust and stable and works at room temperature. There's various advantages and disadvantages to all of these systems. I mean, for example, here the line width is much broader, and the way that the photocycle works means you can't spin polarize into the ground state. But nevertheless, it was pretty cool to see, like, we put DNA into some bacteria, and then later we measured on a pretty much, other than the radio equipment, standard fluorescence wide field microscope, this fairly large ODMR resonance. And so just to get into then how these proteins came about, like what is directed evolution doing in this case? Well, I guess I'll start with what is directed evolution. So originally, we started with a protein, or our collaborators in the US started with a protein that exhibited a very small magnetic field effect. And they took that protein DNA sequence and they applied basically random mutations across it. And they transformed those variants that were generated. So you have a library now of maybe thousands of different proteins that are all like one amino acid different. And you can screen these on a colony, so on a plate, sorry. So you can grow maybe a few hundred colonies on a plate and image the entire plate and apply a magnetic field and see which one is doing the most change when you apply magnets. And then you can pick that colony and extract the DNA from that and apply more random mutations and repeat the cycle. And so after roughly 10 repetitions of this cycle, we are left with the maglev that I presented with this massive change in fluorescence. And so essentially what is being tuned here then are probably these chemical rates. So it's not necessarily changing that much about, I guess, the singlet triplet mixing. But what is changing is these downstream rates. So like how the recombination happens. And there's some good papers showing that like by adjusting these rates, you should see these different sized magnetic field effects. Perhaps a bit less understood is how this translates into different ODMR contrast. And so this is kind of what the measurements look like as you do this directed evolution screening. So with a few mutations on the left, there's a small magnetic field effect. And then as you keep adding mutations to this protein, the magnetic field effect kept growing. And then in the last case, I asked them to select for, if they could select for like the speed of the change rather than the kind of absolute change. And I guess we got this variant, which in some sense kind of saturates very quickly. In another sense, it's slow because actually the slope that it changes at is much smaller because the overall effect is smaller. But the point is that you can, if you decide what you want to select for, nature will oblige and kind of give you a protein that does that. And we also measured the radio field effect or the ODMR contrast for all of these variants. And I guess interestingly, it doesn't seem to be like directly correlated with the magnetic field effect. So one thing that we want to do in future is see how could we perhaps select on the ODMR separately from the magnetic field effect. This requires a different screening setup because you need to now be able to measure the effect of all your variants in ODMR, and just like getting the radio signal to them is a little harder than the magnetic field. So some of the equipment to do this, we have this wide field microscope. It's designed for doing actually far beyond these kind of measurements. So you might see in a minute. So it's, I guess, a little overcomplicated, but basically it's got an illumination source, and then up the top, you can see how we applied a radio signal in this case. So it's a stripline PCB. Actually, we found there's some weird resonances depending how you design this PCB. I mean, this is a general known thing throughout people who do NV physics. You're always trying to apply, for this kind of experiment, the most broadband microwave resonator that you can if you want to sweep frequency. And this is just a little bit of a challenging thing to do, depending how sensitive your system is. We also made this magnetic current driver, basically, so that we could have an electromagnet that could do kind of arbitrary current signals. It turned out that this was maybe overkill. You actually just need to turn the magnetic field on and off. You don't necessarily need arbitrary waveforms between negative and positive fields, but we can do that if that's useful, and some RF system to kind of drive this whole thing. So our collaborators, so this is Christiane Timmel's group and particularly her student Anna, did some more measurements to kind of show that we're definitely looking at a protein, because you can actually take FMN and mix it with like tryptophan and see some of these effects. So we really wanted to be sure that this is like this genetically expressed thing. And it certainly is, because these peaks on the right of this this spectra, this kind of structure tells you that the FMN is actually bound to the protein rather than free floating in solution. And we've also looked at some things like how the radical pair forms over time and what different states the FMN goes through. There's kind of a lot more characterization to do here to really identify the radical pair, identify the backbone amino acid that is participating with the flavin. I mean, it's probably tryptophan, but just kind of establishing that. And yeah, and so since this has come out, there's been some great kind of follow-up works. There's been kind of a flurry of work from different groups. One of them is a group in Germany who showed this working with a similar love domain protein, so another flavin binding protein, and they looked at this pulsed, maybe pulsed in inverted commas, because this isn't like a Rabi experiment or something. It's just changing the microwave driving time, and you can see that even well after the kind of laser turns off, the radio field effect is still, the contrast is still going up. So basically what this is saying is the triplet lifetime is long enough that although the spin radical pair lifetime is quite short, in that amount of time, you kind of create a bunch of triplet states with the laser pulse, and then they're still generating spin correlated radical pairs throughout this 100 microseconds. So interesting experiments you can do like this. Our other collaborators have been looking at the theory side of this. So can we understand more rationally like what these mutations are doing, which amino acid should give us a bigger magnetic field effect or a different kind? And they're looking at things like how the FMN and tryptophan, I guess, rotate within the protein itself, and which needs to be stable and which needs to be kind of more floppy to enable the spin radical pair to be generated, separate, come back, recombine, all of this. And there's a lot of follow-up to do here as well experimentally. And so with that, I'm going to hand over to Harrison to tell you kind of, I guess, more so why we got really excited about this at the start in terms of some applications that we're now pursuing.
Great, thanks, Gabby, and thanks everyone for your attention so far. So for those I haven't met, I'm Harrison. I'm a professor in Oxford in the engineering department, working broadly in the areas Gabby just talked about. So Gabby's nicely explained to us some things about the biophysics of what's going on in these proteins. The next thing I wanted to talk about was a bit about why they're useful, or at least the applications that we have investigated and are developing, and then also some things about what we think might be useful next steps, which we're also interested in developing. So just to briefly rehash, as Gabby showed, one of the simplest measurements we can do here is put a magnet on and off and see a change in fluorescence. This is really simple in the sense that you can have like literally a fridge magnet, or you can pay a couple €100 to buy like a electronic power supply and electric magnet and do this as a retrofit on virtually any fluorescence microscope that exists. So therefore you just are using exactly existing hardware. The first thing that is useful for this, which we're already working with various kind of medical focused biologists to try is things like lock-in imaging. So hopefully everyone from physics has come across this at some point. Basically, the idea is you modulate the signal you care about at some frequency, then you look back and try and extract only signals at that frequency from what you measured. So in this case, imagine I have some messy sample like the one shown here. You can see there's all kinds of fluorescent rubbish here. It would be difficult for me to work out which is the fluorescent protein tag that I care about because of all the other signal there. However, if I would start flashing my magnet off and on and I tag the thing I care about, then I should just see one thing flashing, right? And so I can easily extract from this big messy background, which is the one that I care about. I mean, obviously I can do this just looking at it, but I can also do it computationally and make it much better. And so this is useful in a lot of cases, both situations where there's a big messy background that you want to see something within, and also the case where you might have time varying backgrounds and things which are varying on all kinds of time scales. And you'd like to, again, just get back this one signal you care about. For example, where is my protein or drug that I care about in this tissue? That's one example of using magnetic field effects alone. A second example would be just using them to differentiate between different proteins. So anyone that's done kind of fluorescent protein biology might know that you can sometimes get 3, 4, maybe 5 different fluorescent proteins in a sample and then extract who is who from a picture. But one of the challenges of fluorescent proteins, they have quite wide excitation and emission bandwidths. So you can't really have, 100 of them in one sample because there's just not enough spread in the visible range to do that. But what you could do is have, let's say, multiple green fluorescent proteins and have some of them which flash and some of them which don't, and some of them which flash at a faster timescale or at a smaller magnitude, and therefore extract back multiple signals from one measurement, again, without having to really add much to your system, like literally just putting this magnet off and on, and then comparing both in wavelength and the amount of flashing, the rate of flashing, and then we can kind of combinatorially expand the number of reporters I can have at once to 10 or 20 or something, which is then very helpful for all kinds of biological assays. So far, these examples I've given have all been just using a magnetic field. Perhaps the more advanced and exciting ones are using the ODMR measurements to then do other kinds of localization measurements in this case. And so generically in biotechnology, a common problem that we have is how do you work out where some protein is? And that can be how fundamental science, or it can also be in many applied cases. For example, if I'm developing a new antibody or drug which needs to target a certain organ in the body, I would like to see where that has gone in a tissue. Similarly, if I'm doing a microbiome study and I'd like to see where are the certain bacteria within the gut of a mouse or a human, I would like to also be able to see where they are within some messy tissue. And so generically, it's important to be able to work out where are proteins. And Gabby said this has been done a lot with GFP. But if I imagine doing this with GFP, here's my ridiculous cartoon koala. And let's say I want to target some organ in this koala. The ideal circumstance would be this, that I would just send my protein and I would see the protein is at that place. But of course, if this koala is like a fluffy thing with tissue, it's actually going to look like this. Like the light coming out of this protein is going to go all over the place. I'm not going to be able to really localize exactly where is that protein because of all the scattering through the tissue. And so this is a challenge which is basically unavoidable using existing markers, and it's something that we can potentially solve using ODMR. So coming back to these curves, Gabby previously showed one of these curves, which was essentially like a set magnetic field. Here we have 5 overlaid. The difference between them is you can see on the y-axis there's different magnetic field strengths. And so you can see as we move up this plot, the location of the resonance is moving. And like that blue line is like just a prediction from the theory of where it should go. So you can see it's very predictable. And so what it's showing is that if I would apply a magnetic field gradient here with the smallest magnetic field at the bottom, the biggest at the top, I would expect the location of this resonance to change. And so what I could do is turn off and on my RF field. In theory, I would only observe responses from proteins which are at a certain magnetic field. So let's just explain what would happen here. So let's say I picked like 600 in my frequency here. If I imagine turning off and on my radio, only proteins that are approximately at this magnetic field, so about between 21 and 22, should give me any response. The rest of them, like this one at the bottom, are kind of flat there, so they shouldn't really do anything. And so by doing this, I could then work out where things are based on where they are in my known magnetic field, because of course I can make a really nice model of my magnetic field and it goes through tissues and whatnot. So it's very predictable where that's going. And so if I have my ridiculous koala thing, I shine in some light, I measure the output, but in this case, I don't even need a camera to measure this output. All I care about is the absolute amount of light coming back. I would then be scanning either my magnetic field gradient or my RF field, essentially to move the resonance up and down the koala. And I should only see the flashing when I hit where my protein is. And so the important thing here is that the location of flashing is set by the RF frequency, which is like perfectly controllable with electronics, and by the magnetic field gradient, which is also essentially perfectly controllable with electronics in that I can send that through a tissue. Unlike light, which I cannot just send to an arbitrary point in tissue and understand exactly where it's going, a magnetic field gradient pretty much perfectly goes through tissue, doesn't get scattered or absorbed very much. And so compared to a traditional fluorescence labeling assay, this is allowing me to then work out much more precisely. And so in our work, which I'll show in a second, we can get this within like a fraction of a millimeter, approximately where things are. And so how do we test this? Well, I mean, actually, based on hassling by reviewers, we kind of said initially that this would obviously work. The reviewers said, well, if it's so obvious, you should make something to do it. And so we eventually did. So we started with a MRI coil. So this is something used in rat MRIs. It's about this long, which is tuned to about 500 megahertz resonance. Our rat, in this case, was literally a piece of cast plastic, and we put holes through this plastic so we could then put in signals at certain locations. We then made an optical setup, which is basically just a photodiode. So there's no camera involved in this setup. It's that long tube is like just a long piece of glass to take light out of the resonator. And we just have a photodiode at the end. So the important thing to note is there's no like spatial measurement of the light. It's just like one sensor saying how much total light is coming back. And then a bunch more electronics. So the RF stuff on the left, our LED and system control on the right to sync up everything and control it all. And so if we then look at like a diagram of what's actually going on, I have my sample here, which has a permanent magnet on one side. So I have like a decreasing magnetic field across my sample. I can then shift that using Helmholtz coils, so I can electronically shift the gradient. And then I have a fixed RF frequency. And so then this is allowing me to basically shift the magnetic field gradient and move that resonance, like I said, with the koala. And so what this should do is therefore, and here's some measurements, if I have samples at different positions along my Z-axis of this scanner, I can see basically these peaks, which show me where that sample is, and I can fit to these peaks to work out kind of a central location. That's at the top is just doing two separate samples. I can indeed mix together two samples and then re-extract where things are to then work out approximately how far are these things apart within this sample. And again, to reiterate, at no point did I actually measure like the location of the light arriving or the direction of the light. It's all based on the resonance conditions, which is the strength that it could also go through any kind of other tissue that you can at least get light through. And so as a next step for this, we're currently looking at making this into a full 3D instrument, where the goal would be really to focus on kind of mice and rat models, where there's lots of assays already in drug discovery where you'd like to work out where proteins are going in mice. And this would tell you in 1D, but you'd like to have a full 3D image to some extent. The next application in our paper, which was actually the last figure there, was looking at the local spin environment. And so here we tried a few things and eventually saw that indeed, if you put with a purified protein this time, so not in cells, we could get a change in the MFE as there was more and more of a gadabutyrol, which has got a bunch of unpaired spins. However, if you look at this x-axis, the units are large. Like this is a lot of chemical there. So is this going to be useful to measure things in cells yet? Not quite, but as Gabby said, we're working on engineering these proteins for new and improved sensing of some of these properties, which is taking me to our future development. So in terms of other relevant systems, so we've talked mostly about maglev so far. For people that are following this field, another two papers that have come out quite recently in 2026, there was one like literally last week from Dominic in Germany, which is looking at cryptochrome and also an iLove, which is a different version of the love domain. Another one from a team led by Sean Bird in Stanford, who have looked at various red fluorescent proteins and also in other organisms like nematode worms here, showing they can localize things in a worm. So that's really nice. A next step for us, like I said, is building some kind of 3D system. So on the bottom here is an image of a mouse, which is taken with fairly standard luminescence imaging. As you can see, it's a bit of a blurry mess. And so obviously the bioscientists and medical people who are wanting to work out where things go in mice would like to make that, instead of a blurry mess, just have a spot in the target organ, for example. And so here, again, we have to try and work both on the algorithms and the hardware to build this system. So it's another thing we're investigating at the moment. Another next step, which is, in our opinion, I think many of your opinions, very exciting, is moving from all the measurement stuff I've talked about to control. That is to say, actually making some biological process happen or not in the presence of a magnetic field. And so if we think about our current system, which is like putting light in and getting light out, the input and output of that are not what I want yet. So the first thing I'd like to get rid of is the putting light in step, because I'd like to not have to shine light into my tissue because it's obviously difficult to get it there. And so one way to overcome that is using luminescence, of which various systems exist, which are used again widely in biotech already. So coupling luminescent proteins to things like maglev, people have already shown can give you a magnetic field response. So there you don't have to excite it, you just get back light, although it's in practice much lower intensity because luminescence is generally weak, has some other trade-offs too. And the next step for that is like I said, the output of the system currently is emitting light, but what we would really like is that emitted light to actually be an emitted changing conformation, for example. And people that know the love domains know these have been used as optogenetic regulators for many years, which is basically systems where you turn on or off light and therefore get your protein to bind to DNA or not, and therefore you can turn on or off expression of a gene. And so that is naturally what they do, although some of the mutations that have been made in the process of engineering them for this have, in theory, prevented that happening. The next step for us is to put that back in or fuse them with other proteins to allow that to happen. But then in theory, you could make a protein, which is going to be in practice a combination of several proteins, which hopefully would be inside of a tissue. You would turn on a magnet that would make it start doing something like binding. You turn off a magnet and not. Some of the people Gabby mentioned in California are working on a company related to this called Nonfiction Labs, which focuses on antibodies gated by magnetic fields. And we're more interested for now in transcription factors because that would help a lot of our other work. And broadly speaking, this would then allow you to use this in applications where optogenetics is not so good. So of course, light control of biology is great because you can send light. If I just have a flat sample, I can send it very precisely within a wavelength of light to a certain location, but it's not really very good going into tissue for the reasons I said. In contrast, a magnetic field, I cannot really send to 1 micrometer in a sample because it's just not how Maxwell designed his equations. However, I can send a magnetic field through a tissue and have it not get scattered. So I can very predictably say where it might go at 10 centimeters depth or 1 centimeter depth in some organism. So it has pros and cons. It's not going to maybe fix all the neuroscience challenges, but there's a bunch of problems where it would be very helpful. And so this is our long-term vision. How are we going to get there? Well, Gabby mentioned we do a lot of synthetic biology of other things too. So something we've developed in our lab for some time is a microfluidics. Basically what this is techniques for doing really high throughput parallel assays, in our case in bacteria. So in the top right of this slide, you can see a diagram where there's a bunch of individual cells in what looks like individual test tubes. And so these are fabricated out of kind of silicon wafers with casting plastic. And so each of those test tubes is about 1.5 microns wide. And you can make a chip which has millions of these on it, or in our case, currently about 100,000 of the biggest chips we have. And so you can see here's an image from the microscope. There's like cells growing in these channels and they get swept away by fresh food as they grow. And therefore you can do very high throughput experiments. To give some idea of the throughput, this is a picture of like 200 cells in this system. If we zoom out on one of these chips, we can quickly see it becomes quite unwieldy to millions of cells. But the beauty of this is I could then potentially make 1 million versions or at least many thousand versions of the protein I care about. I could put them all into this chip at once. I could do all of these measurements at once. And then if I can do on-chip sequencing, which has been demonstrated in the last couple of years, I could get back all the sequences. So I could have like 1 million proteins and 1 million of these MFE traces and ODMR traces and all this characterization. I could just get all of that back in one experiment where the conditions are consistent across all the proteins. I can have very strong like low noise measurements and then basically probe huge amounts of the protein fitness landscape to work out exactly how do all these aspects of the structure and design of the protein define what happens. And so in our opinion, this is really a great system for going forward in this field. There's lots of challenging engineering, and we're also building some other apparatus, as Gabby mentioned, to do this kind of experiment. But if you can do really high throughput experiments, so compared to the previous ones, which was like picking one thing off a plate at a time, in our paper, there's like 8 sequences or something, and we actually characterize 4 of them in detail. The goal here is to have like millions of sequences. And then we can start thinking about very high throughput modeling techniques to try and predictively design better proteins and really understand what they're doing. So we're getting towards the end. If you'd like to read more about the science of what I talked about and what Gabby talked about, please have a look at our paper, which also, if you're interested in following any of the methods, we have quite a big detailed supplementary material on. If you have questions beyond that, then feel free to email both of us or either of us. And we'll try and help you. I mean, particularly if you're trying to set up a system like this to measure these proteins in your lab, we're very happy to help advise on that. And then hopefully we'll be coming up in the next years with other papers which are developing new proteins, some of these very high throughput methods I talked about, and then also some of these applications. So if you're also interested in any of these applications, we'd be keen to hear about that. And of course, we're pushing on some of these already, but I'm sure there's many more things that we can't possibly do. We're only one team of a few people. So it's great to have more people interested in this. And we've been happy to collaborate with lots of people around the world that have reached out already. So with that, let me say thank you very much first to my lab, Gabby in particular, who has led this work. Also, a lot of the work here was quite collaborative. So in Oxford, there's Christian Timmel, who some of you may know, who worked a lot and continues to work on avian navigation. So how do birds sense magnetic fields, and lots of fundamental chemistry around that. And also Lewis and Anna from her lab at the time, who contributed to this work. And then JP, who some of you may know from Melbourne, who works more in nanodiamond type areas. And then, of course, Maria and Andy from Calico Biosciences, now partly working in their own companies, who worked a lot on the direct evolution for this. And with that, I think we have about 10 minutes for questions. Thank you, everybody, for your attention.
Thank you. This is quite fascinating work, I would say. The something that intrigued me was, so you talked about your efforts to build actuators into the system. Now, obviously, if you had a way to, say, drive transcription factors, something that would be very intriguing would be to use this to drive a selective marker, which could allow you to do directed evolution to further improve your original sensor, for example, to, I don't know, improve the dynamic range. Have you thought about that? Would it work? What are the limitations? How would you do it?
Yeah, so in our lab, we also do a lot of growth-based directed evolution, which maybe is what you're hinting at with a selective marker. I think this is something that would work at some point. Initially, though, we kind of expect these things to be really bad, in that we expect like fractions of 1% of change of the expression, which is quite difficult to measure if it's the expression of a gene, if you're doing it in a high throughput. And so instead, our current approach is trying to make much faster readouts. So for example, there's various ways of observing dimerization of a protein on like second timescales, things like FRET, for example. And so we're starting off with things like that, where we can then look at fast changes, because then we can make quick changes to the magnetic field, every 10 seconds or something, and do a bunch of off on, and then see which one is responding. And then eventually, if you had a big control system that had a big response, then you could do all of that. And people have done stuff like that for the love domain engineering, but it kind of requires you to start with something that has a reasonable dynamic range, because otherwise it gets washed out in the noise of growth and variability. So there's a lot of nuance to directed evolution experiments. You have to pick the right thing, and often it's not the same thing at different scales or different performance levels of what you're doing.
Thank you.
Yeah, great talk, guys. Well done. So I'm also interested in your kind of ambitions to make an actuator, something that controls function. But the kind of canonical mechanism of the love domain is this: it forms this cysteine adduct, right, with the flavin, and that's what then causes the structural change in the J alpha helix, etc. I'm interested in your thoughts, how you kind of square the circle of that with having a magnetically sensitive radical pair, because that's thought to go via a radical mechanism potentially, but the cysteine is so close that the energy gap between the singlet and triplets will be huge. So yeah, your thoughts.
Yeah, so that is what I was hinting at, where I said we had done some changes which may have made it worse as a regulator. So indeed, that could make it worse. One thing we're doing is just testing if we could make them get reasonable magnetic field effects without making that mutation. The second thing is things like Brett and Fret. So imagine that currently, if you look at this diagram, we're like exciting it around 450 and the emissions low 500 nanometers. You could then put a red optogenetic regulator next to it, and then hope that you get some energy transfer into that, which then has an intact actuation mechanism. So yeah, there's a bunch of possibilities. It's unclear to us exactly what the best one is. We're kind of doing a let's try a bunch of different stuff approach at the moment. But yeah, you're right that there's this current kind of competing factors. And so it's not going to be, I think, as easy as just jamming two things together and it works pretty well. It requires each of these steps to be somewhat optimized.
Thanks, Harrison. So switching a little bit from quantum 4 to quantum in, Are you aware of any organisms that metabolize ASLOV in any way for magnetosensing?
As in, do we think other organisms are using this as a magnetosensing
Yeah,
mechanism?
like animals that eat plants or us or other organisms.
I think not to our knowledge. I mean, it's just a generic protein which is used for a range of things. I mean, LOV stands for light oxygen voltage sensing.
Mhm.
So they are kind of diverse proteins used across many domains of life. The AS is just the AS literally stands for the oats Latin name.
Right.
So yeah, it has a bunch of functions in nature. We're not sure if any of these are particularly quantum, and we haven't really looked into it, to be honest. If it's an interesting question for people, I think it is, but yeah, it's not been our priority.
Thank you.
Okay, I see a lot of questions here, but I will go.
Great talk, Harry. You mentioned that you have as a possibility of application to measure local spin environments, but that there was a difficulties in that direction. Can you elaborate a little bit more of what the difficulties are in sensing that local spin environment?
Well, one difficulty in this existing graph is that it was not very sensitive. Of course, this was purified protein and like a literal chemical floating around in solution. So I guess you can calculate the mean free distance between them, and it's something a bit large. A more interesting thing might be similar to some other talks we've heard about things like Ross sensing. Again, it's kind of unclear to us how sensitive it would be. And something we've started investigating, but again, haven't pursued because it hasn't been our top priority, is how do we make like good perturbations in a cell which make a known amount of Ross and hopefully long-lived enough that we could sense it. So again, I think there's challenges there in terms of like what concentration and proximity is it going to be sensitive to. And I think a lot of them, at least from our perspective of what we're good at, are things like how do we make that perturbation and predictably know what we've done to therefore be confident that our sensor is sensing something legitimate. Because I mean, you just put stuff in a microscope and measure it, you always get like all kinds of noisy mess. But we'd like to precisely know, like have a separate control. And I guess we heard some nanodiamond talks about this. So I think
what
there's
about touching this one to a larger macromolecule that has a very weak magnetic field that is often difficult to sense in ensemble experiment? Is that possible with
so someone in our lab is already working to make them fuse to different ferritin systems in bacteria, but that's not necessarily as a Ross sensing mechanism. But yeah, certainly that would be interesting as well.
Hi, thank you for the interesting talk. Do you, or has anyone looked at, or are you looking at using the directed evolution to induce zero field splitting in the triplet? So having non-zero D&E parameters. And then secondly, if I'm allowed, do you see any orientational dependence in your RF driving? Or is like the principal axis, spin principal axis, like uniformly distributed?
So the first one, no, but if there's nice, convenient measurements to do this, we'd be happy to hear about it because we're interested in doing directed evolution of all kinds of things we can measure. Secondly, no, because currently all of this work is proteins either in solution, where they're in random rotation orientations, or in cells where currently they're also in random orientations. We have thought a little bit, but haven't pursued how we would localize and fix the orientation within a cell. Because like if we come back to this kind of thing, I can at least set the axes of my cell exterior to be fixed, if you look at that. So I can force all my cells and I can see the orientation. And so then plausibly I could approximately fix the orientation of some proteins in that cell, for example, binding them to membranes. I would at least get them in a plane as opposed to complete 3D randomness. But again, this hasn't really been we haven't had a specific question to pursue to do that yet, but we did think about it a bit.
Thanks for the really interesting talk. I think this is a question more for the first part as well, where in the radical pair mechanism, the singlet triplet mixing is driven by the hyperfine, which is driven by the local nuclear environment. And there's some theories that specific nuclei can cause like very strong mixing or strong singlet states. How much do you know how much, or do these amino acid swap outs change the hyperfine environment at all?
So I think we have not seen anything which changes the width of our resonance peak, which is partly determined by some of these hyperfine couplings. We have currently sitting in our lab, but not yet measured, systems to making deuterize proteins and cells, which is, I guess, the next step in this direction.
Thanks.
saw a question here, but I'm not sure if I'm missing somebody here. No, good. Then